Well, welcome back. Our next speaker is Dr. Talani from the University of Cagliari. We'll talk about neurophysiology and the knotty neural networks.
Thank you so much, Paul, for inviting me even here in this great school. And thank you even for Paula to show these great images that make my presentation much easier, probably. Okay, I'm an electrophysiologist from Cagliari. I work in the National Research Council. And I want to talk about electrophysiological tools in order to understand and to study the neural speech in the brain. Let me try to start my presentation from here. The brain, okay, is a knotty neuronal network in which billions of neurons are able to communicate each other with a chemical and electrical signal. But how can we study these fascinating mechanisms? We can use, as Paula showed you, we can use the neuroimaging in order to label the neurons with different colors and localize specific neuron in specific brain area, or use the confocal microscopy in order to study the microstructure like a synaptic dendritic spines that we talk about these spines later regarding the synaptic plasticity. Or we can study, for example, the expression of specific proteins in the brain, like channels or receptors in the plasma membrane. But how a single brain structure work and regulate the neuronal physiologic function, that's why electrophysiology exists. Okay? Let me give you a little points about the story of electrophysiology. It's a long story starting in the 17th century when Isaac Newton suggests for the first time that bodies can operate to greater distance, propagating their signal along solid filaments from organs of sense and to the brain. After him, I want to remember Luigi Galvani, Lucia Galeazzi, and Giovanni Aldini, that they set up for the first time the basis for electrophysiology and predict the presence of ion channel using frog legs. And even Leopoldo Nobili, using frog legs, he measured for the first time the current associated with a muscle contraction. In fact, he called this current frog currents for the first time, or proper currents. Also, Carlo Matteucci and Émile Dubois-Raymond, they clearly demonstrate the biological nature of electric signals and measure what we know today about the action potential in both muscles and nerves. Julius Bernstein, he invented the differential rheotome that allowed to measure for the first time the action potential and estimate the resting membrane potential with about -60 millivolt. And finally, Alan Hodgkin and Andrew Huxley, they fabricated mini electrode, glass electrode, inserted into the axon and performed for the first time direct recording of resting and action potential from the axon of a squid, using the voltage clamp technique. For the first time, they measured the ion currents involved in the generation of action potentials. And ladies and gentlemen, in 1975, we know for the first time Bert Sackman and Erwin Neher that introduced for the first time the patch clamp technique. Okay? A new generation of electrophysiology is born now. They won the Nobel Prize in 1991 in physiology and medicine. Okay? They invent for the first time this technique using practically the same electrode used in the past, the glass electrode. But the genius is that they thought to apply a negative suction in the electrode in order to selectively focalize the suction in a little tiny patch of the membrane, okay? In order to isolate a little tiny part of the membrane with a few receptors in the membrane, okay? Where is the gain? Is here, because after Hodgkin and Huxley, you can see here that before the suction you have a lot of electric noise. So it was pretty impossible to record tiny currents like this one that are generating from the opening or the closing time with a single receptors. So it's a huge discovery. There is the plasma membrane. As you can see, the plasma membrane is a bilayer strata in which you can find some receptors or channels that make the membrane permeable to selective ion currents. Try to imagine the membrane as a simple electric circuit composed by a resistor and by a capacitor. Why resistor and capacitor? Okay? Because the membrane is have a bilayer structure that is able to accumulate at both sides different electric charge due to their bilayer. Okay? And why resistor? Because ions are diffused across the membrane throughout specific channel that acts as a resistors. So if you have a lot of channel opened, you have a low resistant effect in your membrane. Okay? If you have, for example, a Purkinje neuron that is really big, the capacitive properties of the membrane is much bigger than, for example, a granular neuron in hippocampus. So the capacitive properties of the membrane depend directly by how big is the neuron. Okay, I think that Paolo can give you more information about these two laws, but I just want to say a few words about this law. They are considered the milestones for electrophysiology. The first one is a Nernst law that allowed you to calculate the equilibrium potential or the reversal potential for every single ion in the membrane. Okay? For example, the sodium have an equilibrium potential of plus 55. This depends by the fact that the sodium is much more concentrated outside the membrane respect to the inside. Okay? Opposite direction for the potassium. In fact, the potassium have an equilibrium potential of minus 75. Okay? The calcium is a particular ion you can discuss a little bit later. And the chloride ion have minus 90 because it's much more concentrated outside the cell. So given these values, it's possible to have the second law, the Goldman law, that allows you to calculate respect to the single concentration or single ion the membrane potential for a single neuron. Okay? If you want to make the difference between the equilibrium potential and the membrane potential, you can obtain the driving force for a single ion. Let's talk about a little bit the voltage clump theory. Okay? If you have any doubt, any question, just stop me whatever you want. Okay? The voltage clump theory was invented by Kenneth Cole and Howard Curtis in 1936 and allows you to fix the voltage of the membrane at the precise value. Okay? How can you make it? Because you can fix the voltage, you have to measure the real voltage of your neuron, you can fix the voltage that you want, and based on this fixing, the amplifier can inject positive or negative currents inside the neuron, depending on what you are deciding to make. Okay? For example, if the membrane potential is around minus 65, okay, and you want to fix it, it's at minus 80, what you need to do? To inject negative current inside. Okay? Any problem? No. Perfect. So if you apply this technique, so when in the membrane something happened, for example, AMPA receptors are opened, stimulated by glutamate. Okay, what's happening in the membrane?
Change the charge.
Which kind of charge?
Positive.
Sorry?
Positive.
The positive. So what the amplifier need to do?
In order to?
In order to fix the voltage of the membrane.
Negative.
Negative current, right. Perfect. So this is the theory of voltage clump. Easy. Okay? So here you can see a typical current, a real current. So what is really happening in the membrane? Okay? So you have a positive current that are going inside the cell, and in the PC you can see the negative current that the amplifier needs to inject in order to fix the voltage of the membrane to the holding voltage. Okay? So here we can conclude that the red one is the theoretical current that the amplifier needs to inject. The black one is the real current that is happening in the membrane. So you can measure indirectly exactly the same current that are flowing throughout the membrane, throughout the AMPA receptors. Okay? This current is pretty the same because the measurement of the amplifier is made 5,000 times per second. So it's really fast. As you can see here, so why we can obtain this triangular current? Because we have, see here for example, we have different opening and closing time opening and closing behavior for the receptors. Excitatory receptors, inhibitory receptors. So here you can see the current that is exactly the average of 100 of the receptors that are open and closed in the same time, or almost in the same time. Okay, that's why you can see a triangular current. Okay. What about the current clump theory? Okay, with a voltage clump, you fix the voltage and you measure the current. In the current clump theory, you fix the current that you want to inject inside the cell, positive or negative, and you are recording the change in membrane potential. Okay, so the current clump theory, the current clump methods is really useful if you want to study the action potentials, for example. Instead, the voltage clump technique is really useful if you want to study the opening and the closing time of the receptors, if you want to study the current mediated by the receptor activation. This is a classic classic setup for patch clamp recording in slides. Okay, we use slides from rodents. Okay, that we have fresh slides every day. And we need a normal microscope, a differential interference contrast microscope. It's better to have an infrared microscope because the infrared allows you to go deeper and deeper in the slice. We use slice for 300 micrometers, but if you need to patch a neuron in the middle of the slice, you need to be much more deeper in the slice. Okay, so infrared allows you something like this. Then you have to have anti-vibrating table. Okay, never mind. All this is not really important to remember, but you need to have a feedback amplifier, head stage. This is the head stage. It's the body in which you can fix your electrode. Okay, in order to make some recordings. Micromanipulators, because you have to make a really tiny movement, because you have to patch your neuron with your electrode. So you have to be really careful in this operation. Okay? The digidata convert the analogic signal arising from your neuron and convert the signal in digital signals, because you need to see what is happening in your PC, in the computer. Okay? Then you need obviously a PC and the recording chamber. Here you can put your slice that it is constantly perfused with the ACSF, okay, artificial cerebrospinal fluid that we made in the laboratory every day, with saline, the right sailings, and all. Okay, these are the different configuration of patch clamp recording. And it's important to show you something like this because for every different configuration you can have different experimental design and different results. Okay? We can start from the initial part when we have our electrode immersed in the ACSF. And the first step is to go in cell-attached recording. So you can touch the membrane of your neuron, you apply the suction, and here is the first configuration that is really useful if you want to study something that I can show you later. Okay? After that, you can go in the all-cell configuration. So you apply a much bigger suction in order to break the membrane inside the electrode. So from here, you have no communication with the stains inside the electrode and the cytoplasm. But here, you have the direct communication with the electrode and the cytoplasm. In this point, you have the total control of your neuron. Here, not. Okay? So you can go in all cell recording. That is the most common configuration for electrophysiologists. Or you can go in, you can pull out your electrode, you can break the membrane like this, and you can go in outside-out patch. So you have, it's like an all cell, it's pretty similar, but you have a tiny part of the membrane connected to the electrode. Okay? Or from the cell-attached recording, you can pull out the electrode, as we made here. You have the breaking of the membrane here. You have this kind of vesicle attached to the electrode, and you may expose this vesicle to the air or high concentration of calcium, and you have the degradation of the outside part of the membrane. See what's happened. Here you have the outside of the membrane exposed to the outside. Here you have the inside of the membrane exposed to the outside. And that's why we call these two configurations outside-out patch and inside-out patch. So, cell-attached, all cell, outside-out, inside-out. Okay? What can we make with the cell-attached recordings? The cell-attached is really useful if you want to study the physiologic firing rate of the neurons. Okay? So you touch the neuron and you start to record what's happening in the neuron. These are really close to the action potential, tiny action potential that you can record from only this tiny patch of the membrane connected to the electrode. Okay? So why this cell-attached is useful? I want to show you a really recent paper that we published in the last days. And as you can see, we studied the effect of morphine or ethanol in the VTA dopamine neuron. The VTA is increase the firing rate of dopamine neuron if you make something that you like it. Okay? If you smell chocolate, if you make sex, or if you have exposed to natural some natural product, for example. But as you can see, morphine and ethanol are able to increase the firing rate of dopamine neurons of the VTA. The best things is that if you perfuse a substance obtained from the Vitania somnifera, it is a plant. Vitania somnifera, it makes the opposite, as you can see here. So decrease the firing rate of dopamine neuron. But if you perfuse the Vitania somnifera together with morphine or ethanol, Vitania somnifera is able to completely abolish the effect of morphine and ethanol. So we propose this kind of treatment in order to counteract some effect of addiction, for example. Okay? Yeah.
Just a simple question. What does it do by itself?
Sorry?
The Vitania somnifera, does it do something by itself?
By itself, yes. Decrease the
No,
firing
I
rate.
mean behavioral.
Or behavioral?
Yeah. Something about rimonabant, which is like an inhibitor that...
No, absolutely. It's not selective for receptors. So we are still studying in which receptor Vitania somnifera may be active.
Okay.
But probably in the GABA receptors, they act throughout the GABA receptor. We will publish another paper, and we demonstrate that Vitania somnifera is able to potentiate the GABA currents for gamma receptors. So we think that even this effect is mediated by the activation of GABA system in the VTA, or in the area that may control the VTA activity. Okay, but this is really interesting because Vitania completely abolished the effect of morphine and ethanol. So the next step is to try Vitania somnifera in behavioral tests in the animals that are addicted to these two drugs.
And that's what I was asking, because like rimonabant was it's a cannabinoid antagonist.
Right,
and,
right.
It was actually commercialized as a
No more.
right, because it does.
The psychotic effect.
Yeah, you don't want to smoke anymore.
Right, but
you think about suicide.
Right,
So,
right. That's why you stop to smoke, right?
Yeah.
Yeah. No, it's a completely different effect.
Okay.
I think that Vitania somnifera doesn't have any effect in the CD1 receptors, for example. The affinity is really, really low, but the affinity is really high for GABA receptors, GABA A receptors. But this is just to show you that cell attached is a simple experiment that can allow you to publish a paper. Okay? It is really easy because it's not difficult like a whole cell recording. What about inside out and outside out? Inside out and outside out are really useful protocols if you want to expose your tiny patch vesicle to drugs. So if you want to make some pharmacological experiments, just for pharmacology, not just for behavior or physiology of the neuron. So you have a tiny patch of your membrane that you can take off from the cell body or from the dendrites, neuroids, and just test your drugs in this kind of a tiny membrane patch. Okay? And as you can see, here you can see a recording from in the normal ACSF and a recording in a drug that decreases the opening time of the receptors. Okay? Low receptors that are isolated in this tiny patch. The good thing for the inside out, for example, is that you can apply drugs that are active to the G protein receptors, for example, that are inside the membrane. So if you want to test some drug that acts in the G protein couple receptors, you have to use this approach. It's really hard if you use all cell recording, for example. Okay, let's talk about the all cell recording. All cell recording allows you to make everything. Okay, that's why it's the most used configuration. With the all cell recording, you can make a study of voltage gated channels, for example, in order to study potassium, sodium currents. You can study the ligand gated channels, mediate currents, and you can study obviously the action potential, the real one, not what we measure in the cell attached recording. The all cell configuration step by step. Okay, the first step, your glass electrode is exposed in an ACSF. You are not touching the cell membrane. Okay. The amplifier injects a square positive voltage step of 10 millivolts, and from the on low, we know the voltage step. The amplifier can measure the current that can go throughout the electrode, throughout the hole of the electrode, and the amplifier can measure the current step, resulting current step. Okay? But when you touch the membrane, the current flow with much more difficulty throughout the electrode. So that's why you have the decrease in the current flow throughout the electrode and the increase in the resistance of the electrode. It is really useful if you want to know when you are touching or not the neuron. In the third step, you apply the gigaseal, okay, like Bert Sackman and Erwin Nier teach us, okay. And applying the gigaseal, you seal perfectly the membrane in your electrode. And as you can see, no more current goes throughout the electrode, and the resistance is really, really high. That's why it's called gigaseal. In the last step, you try to apply a much stronger negative pressure, and you broke the membrane under your electrode. And from this moment, you have a passive reaction to the neuron, to the voltage step applied, okay? And then you can make a lot of stuff throughout your neuron, because you can control the voltage, you can inject current, and a lot of experiments. Unfortunately, we have a lot of resistance in our patch. So the students, you can imagine that students, when comes in our lab, needs about 1-2 months in order to learn how to patch. Because if you don't have a perfect patch, you have a lot of resistance that can change your membrane resistance. They have to be very close to the real one. So you have to decrease the resistance of the patch, you have to have a good sealing in the patch. The resistance of the pipette is stable because it's a glass pipette. Okay, so the resistance is stable. The seal resistance is a resistance really important because it's the resistance in parallel with your patch electrode and the ground. So you have to take in account a lot of stuff in order to learn how to patch. So Hodgkin and Huxley applied the voltage clump method in order to selectively isolate sodium and potassium channel that are the current that are involved in the generation of action potential. And we made the same thing in our laboratory. How? Just apply the patch clamp technique and the voltage clamp technique, okay? We apply different steps of voltage, starting from -80 to +60. So we cover all the voltage that are covered by the action potential variation, okay? And we record the different kind of currents, okay? You can see the negative part of the current and the positive part of the currents. Which current are they? We apply a pharmacology, and as you can see, blocking these, see the red one, okay? Blocking with TTX, the red one becomes the green one. So you can block all these currents mediated by the red reaction with the TTX. So you can conclude that the negative part of the current are mediated by sodium activation channels. Okay? The blue one is the channels that mediate by potassium activation. In fact, if you use tetratrimonium, it's possible to delete completely the currents. So we made in the laboratory the same that usually Hodgkin made in the past. So it's possible to make it throughout the voltage clamp technique, okay? What about the current clamp? As you can see, with the current clamp, you can inject negative or positive currents, steps, and you can record the reaction of the membrane, okay? So you can study even the passive and the active properties of the membrane. Passive are when you apply a current step and you don't see any action potential. It's the passive properties of the membrane, okay? But when you see the action potential is the active properties of the membrane. And as you can see here, applying -80 current step or +80 current step, you can see no action potential in the negative one, but when you depolarize the membrane, you can see the presence of action potential. For the action potential, we can record, for example, the spike frequency. We can increase the positive current injected, and as you can see, we have an increase in the spike frequency of the action potential. Or you can study the spike latency. That is the latency, the time that you have between the application of the step and when you see the first action potential. Or you can study, for example, the passive property of the membrane. For example, the resting membrane potential, because you know the voltage, because you measure it, you know the current, because you are applying it, and the amplifier calculate the resistance of the membrane. Or you can calculate the V threshold, because the V threshold is the threshold right before the generation of the action potential. Or you can calculate the Rio base. The Rio base is the minimum current that you need in order to obtain the first action potential. And this is just a sample of one of our publication in which we isolate the animals just after weaning, and we demonstrate that we have a decrease in excitability in the neurons of dental gyrus from C57BL/CJ mice. And as you can see, we conclude that there is a decrease in excitability because here you can see the decrease in firing rate when we depolarize the membrane. Okay, and we have the increase of Rio base, so we need to much more current in order to elicit the first action potential in isolated mice. And again, we have the decrease in action potential frequency. It is the control one, and this is the isolated one. And even the increase of action potential latency. Okay? Probably all these changes depend by the fact that isolated animals have a much more depolarized resting ventral potential. Okay? So another interesting study that you can make with the patch clamp recording is to study the effect of neurotransmitter or on ligand gate receptors, spontaneous or evoked release. So you can record at postsynaptic level the spontaneous activation of postsynaptic receptors, probably maybe AMPA receptors, GABA receptors, but that are activated by the spontaneous release of neurotransmitter from the presynaptic terminal, or you can evoke the release of presynaptic terminal, and so you can record the resulting current after the activation of AMPA receptor or GABA receptors. As you can see here, this is a typical recording of spontaneous current, in which you can calculate the amplitude of the current, the rise time, so the time that the current needs to reach the maximum level, the decay time, the time that the current needs to reach from the maximum level, again the baseline, and the frequency, so the number of the currents. Make this kind of simple study. We published a paper here in which we demonstrated that animals that are withdrawn from ethanol consumption have a decrease in the frequency of AMPA-mediated currents. Okay? And this decrease, as you can see here, it's parallel to the decrease of dendritic spines, the long-thin spines, in medium spiny neuron in the nucleus accumbens. Okay, so again, this is a really easy study that can allow you to publish in important papers. This is a typical sample of evoked current. So we need to stimulate at presynaptic level the release of neurotransmitter. And here for this experiment, we calculate the amount of the currents mediated by the activation of AMPA receptors that we can make it clamping the membrane at -70 millivolt, or the NMDA receptors holding the membrane at +40. Because we need to depolarize the membrane, because at the physiological level, NMDA receptors are blocked by the magnesium. So you need to depolarize the membrane in order to have this unblocking of the magnesium and make the NMDA receptor working. Okay, so we can calculate the AMPA and NMDA ratio, because using the same amount of stimulation, you have a good AMPA current and a good NMDA current. This is really important because you can calculate the proportion between AMPA and NMDA in normal animals or in animals subjected to some pharmacological treatment or some animal behavior. As you can see here, in the same paper that I showed before, you can see that during ethanol addiction and during ethanol withdrawal, we have a decrease in the amplitude of AMPA receptor-mediated currents, okay? With no really good variation for the AMPA receptors. So we can conclude that this kind of variation are mediated by the decrease in the glutamatergic synaptic components in the medium spiny neurons of the nucleus accumbens. And this variation depends by the fact that during ethanol addiction and during ethanol withdrawal, you have a shrinking of this kind of submolecular composition proteins, for example, the medium spiny neuron or the dendritic spine density, that depend by the fact that you have no more connection for the dopamine, okay? This is really important because if you want to make some neuronal modeling, you have to know which are the receptors in certain type of neuron that are involved in the control of membrane potential. Because here are current mediated by the GABA-A receptors, but located at the extrasynaptic location in the GABAergic synapses. The extrasynaptic GABA-A receptors are different from the synaptic one because the location, so they are extrasynaptic obviously, the subunit composition, the extrasynaptic receptors are formed by alpha 4 and alpha 6 just in the cerebellum, respect to the alpha 1 and 5 in the synaptic composition. And other synaptic, other subunits are the delta subunits instead of gamma subunits at the level of extrasynaptic receptors. As you can see, the extrasynaptic receptor have a pharmacologically really specific pharmacology. In fact, they are sensible for the GABA. They show high affinity for the GABA, and when they are opened, they remain opened. Okay. So as you can see, if you apply a GABA agonist in this case, you have the shift of the baseline at a negative level, and you have the increase in the noise variance. Okay. And again, we published this paper in which you can see that the isolated neuron have an increase in the tonic composition. You can see the shift applying there is another agonist, but the shift applying the agonist is much more bigger in the isolated animals with respect to the control one. And even the noise variance is much more bigger here with respect to here. Okay? I want to show you just few samples of the papers just to demonstrate that it's possible make some easy experiments to publish and to have a paper ready for publication. So how can we study the excitability of a circuit and not of a single neuron? We have to apply the extracellular field potential recording in slice. So, since now we're talking about just the intracellular recording, the patch clump recording is like a kind of intracellular recording. But we can also try to see what's happened if we put our recording electrode outside the cell. Okay? So in this case, there is the hippocampal formation, there is the dental gyrus, there is the CA1 formation. You can see here the CA1, CA3, and delta gyrus. We have glutamatergic connection from the delta gyrus to the CA3 and from the CA3 to the CA1. So it's really important that you remember this kind of circuit because we can stimulate the glutamate release here in the CA3 and we can record what's happened when the glutamate is released in the dendritic portion of CA1. Okay? As you can see, there is the stimulating electrode. We can stimulate the glutamatergic afferents from the CA3, there is the CA1 neuron, and outside we have the recording electrode. That you can put the recording electrode outside the neuron and not more inside like a patch clump recording, okay? So when you stimulate, you have the release of glutamate, okay? Glutamate can act at postsynaptic level activated the AMPA receptors, like here. AMPA receptor allowed the inflow of positive current, sodium positive current, but if you have the increase in positive current inside the neuron, you have a large amount of negative current that remain outside. Okay? So your electrode that is still outside measure the negative inflection of your recording. And then you have the termination of your signal. Okay? So there is not a current. Okay? There is a typical potential recording of outside the neuron. Of this signal, we can record, we can analyze, for example, the slope. So there is a straight line that approximates as better as it can the deflection of your response. Okay? And you can build the I/O relation in which you increase the stimulation value of your presynaptic terminal, and you record the increasing again the postsynaptic field potential recording. Okay, so you can calculate the AC50, and you can use this response to evaluate, for example, the change in synaptic plasticity. The field potential recording is really useful to analyze and to study the synaptic plasticity. I think that you want to have a break? No, we can. You have any question since now? No? Yeah.
Yes, I don't so well the meaning of the slope.
The slope, the meaning of the slope? Oops, sorry. Okay, the slope represents the straight line is a regression line of your response. Okay, so represent the strength of your synaptic. Much more the slope is inclined, much more you have your response bigger. Okay, so you can construct this AC50 relationship. Any other question? Yeah.
What does EC50 stand for?
The EC50 is a value that you need to use in order to study, for example, the plasticity. So if you want to study if this response may be modulated, it's pretty much impossible to modulate this response. If you want to have, for example, an increase in response for plasticity. So the same question, in the same way, you cannot use this response, because if you apply a drug, for example, that decreases the response, you don't have any possibility to decrease this low response. So before starting a new experiment in field potential recording, you have to make something like this, because you have to see which is the 50% of the activation of the receptors or of the field recordings in your slice. So how can we study the neuronal plasticity? So neuronal plasticity in neuroscience, synaptic plasticity is the ability of a synapse to strengthen or weaken over time in response to increase or decrease in their activity. The duration of this change can be short or long-lasting. The short-term plasticity is a transient phenomenon. The duration is around milliseconds, seconds, not much more, in which synaptic efficacy changes over time in a way that reflects the state of presynaptic activity with a potentiation or depression of synaptic function. On the other way, long-term plasticity is a persistent change of the synaptic function. We know that long-term potentiation and long-term depression are the basis for memory and cognition, for example, in the hippocampal formation. Okay? How can we study the short-term plasticity? Okay? The short-term plasticity is a transient phenomenon in the synapse. Okay? The short-term plasticity, you can study this phenomenon by applying the Pulse protocol. So you apply two different stimulation on the presynaptic terminal, and you can see that even you apply the same stimulation of the synaptic, the second response is different from the first one. How can this is possible? It's depending by the calcium, because if the time in which you are applying the two different stimulation is really close, during the second stimulation you have the release of calcium inside the cell, but you have a residual calcium arising from the first stimulation. And if you have enough neurotransmitter that could be released, you have a much more neurotransmitter released during the second response. Okay? This is called pipe pulse potentiation. Okay? And why, in which kind of synapses you can see something like this? In the synaptic that are low probability of neurotransmitter release, because you have a lot of neurotransmitter here ready to be released. Okay? And the opposite way, you can find something like this, in which the second response is lower than the first one. You have the calcium liberation, you have the calcium increase in concentration, you have the calcium residual between the first and the second, but you have the second response that is less bigger than the first one. Why this one? Yep.
When we say low and high
Yep.
probability of release, can you put numbers on it?
Sorry?
What's the number value, like lower than a half or?
Oh, no. It's just lower. We don't have an exact value. I just put here like, just like the samples. Okay, don't worry about the values, but you can have 1.08, 1.1, 1.9, 2.5.
That's not what I'm asking.
Okay.
I'm asking about the probability of release. Is
Yeah.
it something like 0.5? Is it something like 0.8 for low? Whereas high is something like 0.9.
Oh, okay, I understand. If you have low probability, the ratio has to be much more high than 1.
Right.
Right.
But what's the number for the probability?
We don't have an exact number for the probability. Yeah. You can just conclude that if the ratio is much more bigger than 1, you have the low probability of neurotransmitter release. Okay, because you have the plasticity, but you can include this kind of reaction for the low probability of neurotransmitter release. Okay. In order to demonstrate that there is a form of short-term plasticity, you have to increase the time of stimulation. You can see a really high effect if you use 50 milliseconds between the two stimulations, but if you increase the time between the stimulation, you don't see anything. Okay? This may help to answer to your question.
Maybe.
Okay. Another form, okay, I can just show you that we use this kind of protocol in order to demonstrate in the same paper that I showed before, that parallel to the decrease in glutamate release in ethanol withdrawn animals, you can see the increase from the low probability, from the high probability to low probability of neurotransmitter release. So we used the pipe pulse protocol in order to demonstrate that if you see the low frequency in the ethanol withdrawn animals with respect to the control, you can see the transformation from high probability to low probability of neurotransmitter release. There is another form of short-term plasticity. There is a form mediated by the activation of CB1 receptor in the presynaptic terminals. Okay? How can you do it? We have to depolarize the postsynaptic terminal for 5 seconds, and we record, for example, the spontaneous activity, spontaneous current activity. We depolarize the postsynaptic terminal for 5 seconds, and after we come back to the normal membrane potential, you see that we see the decrease in amplitude and frequency of the currents. Okay? So just because during the depolarization we increase the calcium concentration at postsynaptic level, and we start the machinery that product the endocannabinoids that goes retrogradely in the presynaptic terminal, activate the CB1 receptor and decrease the release of neurotransmitter. Okay? This is called DSI if you are studying inhibitory synaptic, or DSC if you are studying an excitatory synapse. How can we study the long-term plasticity? You can use the Ltp protocol or LTD protocol. Okay? Using the Ltp, we record with the AC50, the field potential with the AC50, and then we apply a train of stimulations in the presynaptic terminal. This train of stimulation induces a high release of glutamate from the presynaptic terminals. And this high release of glutamate may activate a lot of AMPA receptors, depolarize the membrane, and activate the NMDA receptors. This activation of NMDA receptors cause a high presynaptic calcium and postsynaptic calcium much more than 5 millimolar. So a lot of calcium concentration at postsynaptic level, activating the PKA or cyclic IMP, activation of nitroside kinase in the presynaptic action because it goes retrogradely and increase the glutamate release, activation of genic expression of new AMPA receptors, and then new synaptic spines and maturation. On the opposite, we see the LTD formation because we stimulate the presynaptic terminal with a low frequency stimulation. This kind of low frequency stimulation make an effect that is the opposite of LTP, because you can make exactly the same activation, but you have the moderate presynaptic calcium and postsynaptic calcium, less than 5 millimolar. This activate the phosphatase that are much more sensible for for calcium rising at the postsynaptic level. And so you make exactly the opposite reaction that we can find in the LTD formation. So this is the difference between Hebbian and non-Hebbian LTP. The Hebbians require simultaneous pre- and postsynaptic depolarization for this induction. For example, you can find the Hebbian LTP in the Schaffer collateral and C1 in the hippocampal formation. But you should have in the same brain area the non-Hebbian LTP. There is a type of LTP that does not require such simultaneous depolarization of pre- and postsynaptic cells. An example of this occur in the mossy fiber from the dental gyrus to the hippocampal formation in the CA3. So you can see here a classical experiment to induce LTP. Okay? You can see the black one is the control condition in which we can stimulate every 20 seconds with the AC50 of field potential recording, and after that we apply the high frequency stimulation. As you can see, after the high frequency stimulation, we see the increase, the dramatical increase in the amplitude or in the slope. We can measure both in the slope of field excitatory postsynaptic current potentials. Okay, but after the increase in the slope, you have a little decrease in the early phase of the LTP, but then the values remain stabilized and higher than the control. So this is the form of long, the LTP, long time potentiation. Okay? If you block with the AP5 the NMDA receptors, you can see the formation of early phase because you have a lot of AMP activation, but after that you can come back to the baseline because you don't have the NMDA receptors activated during the process. Okay? So this means that this effect is mediated by the activation of NMDA during the plasticity. Okay? And here you can see a scheme that I can show you. and I can give you. And here you can see with this kind of publication is that after LTP formation, you have an increase in the formation of new dendritic spines in the neurons. Here you can see the induction of LTD, not of the LTP, but it's pretty common, the same reaction. We have opposite reaction, we have several function of the LTD, several involvement. For example, some publications suggested that the LTD is mediated by the activation of endocannabinoids signals in the neurons. But as you can see here, after a stable recording of baseline, after the application of low frequency stimulation, we have a decrease in the response of our field potential. And this decrease remains stable and low with respect to the control during the time. And here you can find a sample of some of our publication in which we study, for example, the LTP formation that is decreased in animal exposed to social isolation. And this animal have also some impairment in cognition. If you put the animals, for example, in the water maze, they perform much less than the control animals in the water maze. Okay? And here we saw even this is the LTP formation, and in the paper that we evaluate the effect of ethanol withdrawal in rats, you can see that during ethanol withdrawal it's pretty impossible to induce a good LTD in the rats. Okay? All these changes are reversed if you treat the animal with L-DOPA, just to demonstrate that the dopamine, the lack of the dopamine, these animals are the principal cause for this kind of impairments in the nucleus accumbens. Okay, I want to show you some different example for using the patch clamp technique. This example is the use of mechanical dissociated neuron. We use a lot in our laboratory. You can dissociate the neuron, you use the slice, and you can use this kind of electrode. So you have just melting the glass electrode that you can use for patch clamp recording. You melt the glass and you obtain something like this. And you have to put this electrode very close to the slice and activate a vibration, as you can see here. And you can dissociate some neurons from the CA1. I don't know if you can see very well here. There is the CA1 of the hippocampal formation, and here we have the dental gyrus. Okay, so you can dissociate some neurons. One you can see here, another one here. It's not really good, but it's really live neuron. This one, this is the CA3 neuron. Okay, there is a neuron from DLA. Just to demonstrate that it is possible to dissociate a lot of neurons in several brain areas. If you have a slice, you are good researcher, it's possible to dissociate a lot of kind of neurons. And here you can see a paper that we published in 2008 in which we demonstrate that the endocannabinoid and guard-dependent short-term plasticity depression in isolate neuron bottom preparation from the hippocampal CA1 region. So we measure the synapsin, the CV1 location. So you can make a lot of different kind of experiments in this dissociated neuron. Okay. There is a video just to demonstrate the technique, but never mind. Okay, we born in our laboratory just for the electrophysiology laboratory study the expression of GABA-A subunit in the oocyte of Xenopus levis frogs. So it's possible to express different kind of GABA receptor in oocyte frogs and just apply the voltage clamp technique in these oocytes. Okay. The good thing is that you can express typical GABA-A receptor that you want. For example, you can express GABA-A receptor containing alpha 4 instead of alpha 5, instead of beta 2 or gamma, and then you can apply your pharmacological experiments in those receptors, in those oocytes. In the last part of my presentation, I want to show you the new frontiers in brain science, the new frontiers in electrophysiology too. It is the optogenetic. The optogenetic, that's this new discipline in brain science that merge optical imaging, protein engineering, and genetic dissection of neural circuits to optically monitor and control neuronal and brain activity with high spatial and temporal precision. So it's possible to make express in your neuron, in your specific neuron, such receptors, because throughout a viral infection, there is a virus, you can inject the virus directly in the all animals or directly in the brain area. This virus has some cDNA that make express in your neuron typical receptors in your plasma membrane. These receptors are sensible to light. And today we know different kind of receptors that should be channelrhodopsin receptors and halorhodopsin receptors. That you make express both kind of receptors in your neuron. The good thing is that the channelrhodopsin receptors are permeable for sodium, and halorhodopsin receptors permeable for chlorine ions. So it's possible just flashing your neuron with the particular wavelength of light. It's possible to activate your neuron or deactivate your neuron. As you can see here, for example, we may express channelrhodopsin receptors only in motor cortex, and when the blue light is on in the head of the mice starting to move around the cage. Okay? Because it's possible to selectively activate those neurons in the motor cortex. Okay? Another sample, really interesting sample, is this one, because we made express the channelrhodopsin receptor channel expressed in the lateral hypothalamus in order to stimulate feeding behavior in the animals. And as you can see here, when you see the lights on, the animals start to feed. When the lights is off, the animals stop to feed. Okay. Now the lights is off, the animals start to running away. But again, when the lights is on, okay, now the animals start to feed again. Okay, this is really interesting because this kind of experiment you can make even in slice. Okay, so it's not only in the old animal. Here, for example, we activate with the blue light with the 20 Hz. These are recording in the VTA neuron. When you activate the lights with the channelrhodopsin receptor expressed in these kind of neurons, you see the increase in the firing rate. Okay, and you see also an increase in the currents mediated by the dopamine release from these neurons. In the opposite direction, if you use this animal but use the different wavelength of light that activate the halorhodopsin receptors, you shut down the firing rate of your neuron. So it's possible to use light and not the electric stimuli. It's possible to control the activity of your neurons. Okay? And just finish. So if you have any question, I'm really happy to give you the presentation, to give you my mail. Just contact me if you want to make a period in our lab. Just contact me. I'm happy to help you. Faster?
No.